So welcome everyone to the Why It Didn't workshop. Nicholas, Lisa, and I organized this workshop because we wanted to give an outlet for publishing good null results in ALife. When there is an unsolved scientific question, most people will try to solve it by trying the most obvious solutions first. And if that doesn't work, it's often difficult to get your lack of results published anywhere. But if you cannot share that experience with others, then every person working on the same question will have to waste time testing the same obvious solution and realizing it doesn't work. So we are hoping that from now on, when you try something that many other people might try, and if you figure out why it doesn't work, you'll consider sharing it here. We didn't have many submissions this year, so we're open to suggestions. So the program is that next we will have a recorded talk by Nicholas Guttenberg and Nathaniel Virgo titled No-Go Theorems in Chemical Heredity. Then a talk titled The Environmental Discontinuity Hypothesis for Downsampled lexicase Selection by Ryan Baldy, Thomas Helmuth, and Lee Spector. And then we will have an open discussion.
All right, so I'll go ahead and share Nicholas's video.
Hello, and thanks for giving me a chance to speak at the Why Didn't workshop. I'm sorry I couldn't be here in person. And what I'd like to talk about has to do with, well, we proposed this workshop, and I think it was a bit of a, not just negative results, but also having a positive explanation why the result turned out to be negative, and that was a bit strange. So I want to give an example from some work that I did at LC with Nathaniel Virgo in chemical heredity. And I want to talk about this in terms of something like a no-go theorem. And what a no-go theorem is a proof that at least there's some particular kind of thing that can't happen. Like in physics, you can't go faster than light or things like that. And kind of looking at that as at least one possible way to utilize a negative result to drive a better understanding of the space. So the idea that we were looking at is chemical evolution. And for evolution, what you want is heritable variation along with some selection. Selection is pretty easy because anything that creates some differential stability or whatever will give you selection. Variation is usually easy to produce because the world is noisy. But actually inheriting that variation is the hard part. And the core of that is usually the idea of replication. So for a biological organism, that is a cell building a copy of itself, and the DNA is the inherited bit plus methylation and so on. But you make one change to the DNA, that change goes to the offspring plus some mutations. Do you have something like that in chemistry? So replication in chemistry is actually pretty common in nature, and you have autocatalysis is the basic form of it. So if you have something like fire, well, fire makes more fire is kind of the simplest form, but you can also have it at the individual molecular set level where you have some set of molecules, which I have in my diagram in red, which kind of collectively, each and every one of them has the ability to produce more of the set from some food source. So you have some food set B, and the red things A are eating that B or are transforming that B into more A. And the A is not being used up. It's actually, it's producing more of itself. So the amount of that in the system grows exponentially over time until it hits some capacity. There's also things that are produced as byproducts of that, which it's not in the equation, but it's in the diagram, which would be called the periphery. And these green things are some decay byproducts or dead ends or whatever. But those also will appear to grow exponentially if the red stuff is growing exponentially, but they're driven by it. So if you got rid of all the red stuff, the green stuff wouldn't be able to get it started again. It would be dead, essentially. And this kind of stuff, there's chemical networks, models of Titan's atmosphere that show that you would have autocatalysis, and it's responsible for the kind of tarry gunk that people think is in Titan. There's even theoretical results that this kind of thing is inevitable, that if you have a big enough chemical network that has catalytic interactions between things, then you will see autocatalysis somewhere eventually. That's a result from Kaufman. So, maybe we have it, and we wanted to kind of understand if that was the case. Telling you what I'm going to tell you, in the end, autocatalysis, yeah, we can get it in artificial chemistries, but what we didn't find was heredity. So, replicating chemical sets are common, but, is that the same as remembering which replicating chemical set you actually had? So, if I have something where I have some, in this diagram, blue and red growing and forming more of that particular kind of assortment versus the green and purple one that they replicate themselves. So the system remembers which one I had. I don't just have the one bit of information, do I have a replicator? No, but I also can remember which replicator. And the reason the diagram is these little clusters rather than a big chemical network is that there's this model of compositional heredity called GARD, where you have lipids in vesicles that pull other lipids in from solution to grow the vesicle, and the vesicle splits. And they've shown that there are multiple attractors in those systems, so you actually can remember which composition you started with. And they can see some evidence of evolution or adaptation. So we wanted to know if this was happening in chemical systems or artificial chemical systems simulations, and we made a kind of a metric to understand that. And the core idea of this metric is that if I have these replicating attractors, then I will have a bunch of clusters, each of which has a fairly small radius in the sort of space of possibilities. And the space here is the space of compositions. But between the clusters, I will have a lot of variance. So if I do many, many runs, sometimes I'll see one cluster, sometimes I'll see another. It'll look like I have a big variance. But if I look inside a run, I'll have a very tiny variance. Whereas if I just have a chemistry that's sort of open-ended and everything is being made and it's all spilling out into chemical space, I might have a high variance, but it's going to be kind of uniform. And every run will give me a similar thing. And so what we did is we collected data of a bunch of runs and do PCA and look at the eigenvalues. And the eigenvalues, when normalized, tell you what fraction of the variance in that data set is explained by that eigenvalue, or the cumulative sum of the eigenvalues tells you how much of the variance has been explained at that point. So if we have replicators, as we add more runs to the data set, the variance doesn't go up because the attractors have whatever distance they have, but the number of attractors doesn't change. Whereas if we have this kind of open-ended exploratory thing where I'm just spilling out into chemical space, the variance will go up as I add more runs. Or I'll see larger distances be represented. So there'll be more things that have to be explained in the PCA. And so what will happen is the set of eigenvalues that correspond to the between attractor distances will kind of form this shelf, and then the other stuff will kind of fall off. And if we detect the location of the shelf, we can kind of count the number of eigenvalues belonging to that, and that tells us the number of attractors. And we can use the PCA components to even to visualize the dynamics and see that they are attractors. And when we fit this to GARD results, we see the same stuff that they saw using another method. And we were able to sort of measure the number of heritable states, and it was consistent with the number of attractors that they saw. And the scaling laws that they saw were also replicated. So this isn't the thing that didn't work, but this is just a tool to analyze the artificial chemistry. The reason we want, or we're not done with GARD is, well, in GARD, these lipids have to have been produced somewhere else, and GARD doesn't say anything about that process. So if you have this, very rich food supply with a lot of diversity of things, and they have this kind of assortment dynamics, then yeah, you can get evolution in that. But would it work if the lipids had to actually be produced? Or even if we're not talking about lipids, if we just had abstract chemical compounds, will you see the same stuff in that kind of chemistry as you would in GARD? So the kind of template for our random artificial chemistries, we had a bunch of compounds with random energies, and we create n random reactions of the form A plus B goes to C plus D. And A and C or whatever can be the same compound. So you could have like A plus B goes to 2C, but they always have 2 in, 2 out. And we kind of ignore the out of equilibrium things of the rate constants, because when we analyze for the stability of these different growing autocatalytic sets, they don't depend, whether the set grows fast or slow, or whether it beats the rate of decay is going to depend on the non-equilibrium constants. But equilibrium considerations will tell us like which things are actually autocatalytic attractors and which things are not. So we set all those rate constants to 1, and then we just have the equilibrium constants that are determined by the energies and by a choice of temperature, which again, we can just set to 1 essentially. So if we run these things and count the attractors using our method, we see 2 or 3 or 4 attractors sometimes, maybe more, but it's not that many. I wouldn't want to try to do some evolutionary search for a complicated problem only having 8 solutions that I could possibly have ever. It's very unlikely that the thing that I'm looking for will be in there, and that evolution is going to be quite closed. It's not unlimited heredity. It's a very, very fixed heredity. But we only look at a couple 100 compounds maybe at the start, so we could just make it bigger. A lot of the times, bigger is different, more is different. So let's scale the number of compounds and see how the number of attractors changes, and no dice. This is the kind of core negative result. And now the rest of this talk is going to be about what we can do given that we saw that. So we could have just tried to tweak it and make it work and find scaling that way. But instead, there was this result from Nathaniel around that time about first order autocatalysis and proving that there could only be one attractor where all of the concentrations were non-zero. And first order basically means that you have things like A goes to 2A, but you don't have 2A goes to 3A kind of reaction. So you don't have anything where the thing that the autocatalytic set's concentration determines the rate of growth quadratically or higher. Or even if those things are present, it's like the theoretical result is that that's the only thing. But also if you have dominant first order, that could basically do this as well and suppress any kind of higher order stuff from mattering over a region of the space. But the basic idea is that if you have only these first order things, you can linearize the system exactly, and you can look at the spectrum of eigenvalues of the dynamical matrix that kind of tells you how you get from one set of concentrations to the next. And because concentrations have to be positive, you know that the Perrin-Frobenius eigenvalue is the highest positive eigenvalue. That's going to be the thing that will be your growing autocatalytic set, and that has to live in the center of the simplex. And everything else, if you want to have that not have a problem with potential negative concentrations, some of the elements of the eigenvector would have to be 0. So you'd have to be either on the boundaries of the simplex, which would be unstable to any small non-zero amount of the other thing entering the system, or you have to be at a single unique point in the center. You can't have multiple attractors in the center of this away from the boundaries of the simplex. And that's kind of a no-go for what we were trying to do, because it says like, well, not even like 2 or 3 attractors, we should only be seeing 1. And if we were seeing 2 or 3, it must be, well, either because of some nonlinearity that we lucked into, or because we were sitting right on the boundary of the simplex in terms of some things that just were never produced by that chemistry from one initial condition, let's say. So, we can understand this. We can also look at this in a more geometric fashion, look at these chemical networks and understand maybe, why is this the case? Is this just a mathematical thing and some weak nonlinearity will solve it for us, or is this something more fundamental? And the issue is, well, if you have a very connected chemistry, let's say we have these 3 autocatalytic sets here, those are the things in red, and there are 3 independent sets. So in principle, what I'd want to get my evolutionary, my heritable variation is I would be able to have each of these either be on or off. So I would have 2 to the 3 possible genomes in this kind of system, 8 possible genomes. But because they produce things in their periphery, if the byproducts of running the set can possibly ignite another set, I don't really have 2 independent sets anymore. So like if I take the set that's on the left of this diagram, that one produces this green thing as a byproduct, and that's going to ignite downstream the one in the middle, and the one in the middle will ignite the one in the bottom right, and the one in the bottom right could ignite the one on the left. So actually, I only have 1 autocatalytic set here, even though I don't have direct reactions between these sets. And just having some extra random reaction take a byproduct to ignite some other set would be enough to merge those sets together, at least in one direction. So there's this tendency, if the more reactions I have, the more sets are going to kind of coalesce and form bigger sets. And that looks a lot like a percolation transition. So this is a picture from Wikipedia about directed percolation. And it gives the basic idea is that if I have some nodes and random links between the nodes, I can ask, like, if I drop something at one point and then populate everything downstream of it, what does the connected sort of cluster look like? Is that going to be a small cluster of finite size? Is it going to be most of the system will belong to that cluster? Is it going to be a distribution? And in a percolation transition, as I add more links, I go from basically having these very isolated clusters to having, I hit a critical point where I start to see the scale-free distribution of clusters at every size. And then if I add even more links, I tend to fall into sort of one big connected component of the system. So the good news is this is a critical point transition. So if this is what's going on, there will be a special value of the density of links such that the number of large clusters goes to infinity, and we could actually have an infinite number of independent attractors to the system. The bad news is you have to hit that density perfectly, and if you're tuned away from it, there'll be some saturation point where no matter if you make the system bigger, you won't get more independent attractors. And the worst news is even at the critical point, the number of large clusters only grows logarithmically with the system size. So even if I scale it up by a factor of 10, I only add one more cluster. I scale it by another factor of 10, I add one more cluster. And that means I'm not going to have a very evolutionarily rich space from this kind of model, even if I can technically get the number of attractors to infinity by sitting right at the critical point and scaling up to a really huge system. In practice, I'm just not going to see it. And I won't see very interesting evolution, or, well, I'll see very limited evolution. It might be interesting within those limits, but it'll be very hard to scale it or make it open-ended. And so that's sort of a no-go result for what we were trying to do. We had a system, and we thought that this would be even better in some sense than GUARD, because we were adding chemical reactions, and this would create all these new opportunities to create different kinds of attractors and get richer evolution and so on. That's what we kind of expected and we hoped, and it didn't do that. And we could have, scaled it or tweaked it or whatever, but instead we had this opportunity because of Nathaniel's result to understand more formally why it didn't work, and in fact why it couldn't work the way we were doing it. We found a no-go theorem about this kind of system. But it's not just the pessimist, oh yeah, this will never work, give it up, because that no-go theorem depends on certain things. And that helped us narrow down what directions to go in the future. So we can look at all of the stuff that we assumed to get a provable this won't work and change those things and see if maybe that opens things up, or maybe it still doesn't work, but that's a direction to go in. We also know that certain things that might have occurred to us to try won't change the fundamental reasons it doesn't work. So we know that we assumed random links, but if we added some kind of finite scale correlations between the chemical reactions, it wouldn't really change the global scaling of the percolation transition, right? If nearest neighbors or whatever are correlated, that just changes the cluster size, but it doesn't actually change the fundamental scaling laws behind the transition. We need to change specific things, and we can not worry about trying the experiment on every single thing that occurs, because if it wouldn't change a percolation transition, it probably won't change this. So, the things that kind of fall out that might work, we were assuming first order catalysis in the analysis, but it's probably not just enough to add some nonlinearities, because the tendency of order by order things is that the lowest order stuff tends to dominate. So it might be that we could get some additional attractors by going to having a handful of quadratic things, but we probably need not just to have some higher order things, but to have the first order things have zero coefficients, or to be very, very weak, so that it's dominated by the higher order dynamics, and it's not just going to be covered by one really fast growing component that ignites everything. So that's one direction. We also didn't consider inhibition. And this sort of everything coalesces into one big cluster. It works as long as everything can be together in the cluster. But if the activation of one cluster guarantees another cluster can't be active, then that story of fire flowing along links and igniting everything breaks down. That big connected cluster is no longer stable. And this also moves away from the kind of analysis that you get this Perrin-Fabini's eigenvalue from. So that is also supported in the sense that adding inhibition, strong inhibition, would really fundamentally change the story, and it might give us more attractors. We also were assuming a very random structure of the chemical space, and the connection to percolation lets us know exactly what kinds of non-randomness are likely to change the story. Specifically things like symmetries, long-range structure, not just chemically local correlations. We even looked at one where we said, okay, we have a rule now that reactions can only have reactants and products that are within a very narrow energy band of each other. And it turns out that that symmetry doesn't make it so you have lots of clusters everywhere, but it does change the scaling law at the critical point from logarithmic to linear, which is a big deal. Because that means now you don't need to go nearly to as big of a system to get a large number of attractors. You still do need to hit the critical point accurately, though, so it doesn't save you from everything. And then finally, all of these things we did in the continuum limit. And if you have small numbers of molecules, there are discreteness effects. So if you're talking about like replicating citric acid or some small molecule, you're likely going to have, you know, millions, billions, 10 to the whatever 12 molecules of the stuff. And so you are going to be in this continuum limit where all of these sort of smooth dynamics matter. But if you force things into small parcels, or if you're using very large replicating molecules, so you only really get to have 0 or 1 or maybe 2 of the replicating molecule in a given place, it's much easier to land on that simplex boundary, even if you have linear autocatalysis. And so you're much more likely to get a large number of clusters. And in fact, you do see effects like this in GARD, where if you make the vesicle size small, you get more attractors. So in conclusion, I think it's useful not just to publish negative results, but also to talk about why those results happened. Not just, I tried this, it didn't work. Because if you just try something and it didn't work, it's very easy for someone else to come along and say, okay, well, I don't like the way that you did it. I think I could do it better. I'm going to try this variation or that variation. And you don't save, like, it's not like it's an inherent good to tell people don't do the thing you're interested in or anything like that. That's not what I'm saying. But it is good to be able to say, well, okay, there's this entire section of the space where maybe, you know, you can save time and not look there, because we have a way of taking one result and have it cast a shadow, so that we know it, you know, that there are more productive places to look. And even a weak no-go argument can help give you this kind of guidance. In fact, it may even be better if it's weak, in the sense that, as opposed to like just not doing it, in the sense that the places where that argument depends on assumptions tell you things, give you directions to try to change, to try to make something else that works in the future. And it's useful to adopt many perspectives, because again, it's very easy to be very, you know, if you take this stance, you can be very pessimistic and so on. Instead of saying, you know, I will prove that it can't work, that's one approach. You could also say, okay, because it didn't work, I'm going to try, before I just scale it or before I just try this other thing, I'm going to say, I need to have some kind of proof that this should work. I'm going to look for a place where a scaling law does happen. And then the absence of that scaling law, in something you can use that to say, okay, I'm not going to just try that over and over again until I have some way of arguing that like this at least must work, or this at least must happen. But you know, maybe that won't work on a given system. Maybe that's not accessible. It would be really hard to prove anything. And then you can try to look for the negative, or you can try other methods. So yeah, it's just, I think there are a lot of ways to extract utility out of these, out of things that didn't work. And I think that it helps provide guidance for directions to try in the future, so that, you know, you can actually conclude things or be done with them, or even just focus on the things that are much more likely to do something different. And again, you know, thanks for letting me have a chance to talk. I'm sorry I won't be here to answer questions. Hopefully, if any of them get posted, I'll take a look afterwards, though. And enjoy the rest of the workshop.
All right, so as noted, yeah, Nicholas isn't here because of time zone issues. So, yep, great to see that there's at least a question from Hiroki. Slack is a great place to post your questions if you have any, so go ahead and do that. So yeah, next we will have our second presentation. So I'm not sure, Ryan, if you're the one that's going to be presenting or someone else, but you can go ahead and share your screen and all that.
All right, can everyone see my screen? Can I start the slideshow? Everything good?
Yes, we can see
All right,
the screen.
cool. Thank you very much. Okay, so hello everybody. I'm going to be presenting a talk called The Environmental Discontinuity Hypothesis for Downsampled Lex Case Selection. And this is work done with Thomas Helmuth and Lee Spector. So first off, before we talk about downsampled Lex Case Selection, we should talk about regular Lex Case Selection. So Lex Case Selection is a selection strategy in evolutionary computation. So it's just a method to pick the parents that are going to make it to the reproduction stage. And also you determine how many children each parent gets. So these are both parts of the selection. Now, part of Lex Case Selection is also how to evaluate the individuals and figure out how good they are. So a lot of methods use a fitness function, but what Lex Case Selection does is instead it uses the individual performance on each thing that you're trying to optimize for. Now, it was initially designed for these things called uncompromising problems, which are essentially problems where you can't compensate for bad performance on one task by having really good performance on another. So an example of this is a calculator. So let's say you're trying to evolve a calculator that can do all the functions that you want a calculator to be able to do. You want to be able to multiply, add, you want it to be able to divide, you want it to be able to subtract, all these things. Now, if you were a calculator that can multiply perfectly, can multiply any two numbers completely with no issue, but it can't handle division by zero correctly, that still does not count as a correct calculator. And so we don't really want to optimize for being able to do all this multiplication if you still can't do this division by zero correctly, and you can't handle it correctly by throwing an error. So the way that Lex Case Selection works is instead it considers each test case as a specific thing that you're trying to optimize for, and it takes them in a random order and selects parents based on who is elite in that random order. Now, we'll show an image in a second that'll make this a bit more clear, but the things that are important is that it usually selects parents with a very large test set, and by very large, we mean usually in practice more than 5, but in this work where we experiment with things around 20 to 200, so in that range. And also there's no aggregation. So aggregation has been found to just cause a loss of information when you're evaluating individuals. And so if you aggregate their fitness scores across multiple different tasks, you end up losing a bit of information about how well they performed and which tasks they performed well on. So again, Lex Case Selection does not do this aggregation procedure. So the way Lex Case Selection works is as follows. Let's take an example here where there's 5 individuals labeled N1 through 5. And there's 4 test cases labeled 1 through 4. Now, again, these test cases in genetic programming, which is the topic of today, are input-output cases. So for example, you say, if you're evolving a function that can tell you if a number is even or not, the input would be a number like 2, and the output would be a Boolean like true or false or whatever. So that's one test case. Test case is one input-output pair. So here there's only 4. And we evaluate them in a random order. So in this first Lex Case Selection procedure, we evaluate them in the order T1, T3, T2, and then T4 afterwards. And so the way it works is we go through and evaluate all these individuals only on test case 1 first. And then the people that make it through test case 1, and by make it through, I mean have an elite performance, so be the best out of the 5 on test case 1, make it to survive to the next step. So then let's say in this case, it was only individual 4 and 5 that made it to this next step. Now, next is test case 3 in the population. And so individuals 4 and 5 are then evaluated on test case 3. And let's say they both tie on test case 3. So they both get the elite performance out of this subgroup here. So they both make it to the next stage. And that's where test case 2 is used to break the ties again. So then now we have here individual 4 beating out individual 5 on test case 2. And so at the end of the selection event, individual 4 is selected. Now notice here that task 4 was never evaluated on. So it doesn't matter how well these individuals performed on test case 4, individual 4 was still selected, and you don't even need to think about test case 4. This is another selection event with the exact same individuals and the exact same test cases. So no differences in the test cases. The only difference is the order in which they're evaluated. So here we look at test case 3 first. And it just so happens that on test case 3, individual 3 is the best out of the 5, and there's no ties. So individual 3 makes it to the next step. And since there's only one person, that person is selected. So individual 3 is selected. Now notice here, 2 identical selection procedures identical in the sense that they have the same individuals and the same test cases, selected 2 different individuals, individual 4 and individual 3, just based on what the ordering of the test cases are. And then here's the final one where we evaluate on test case 4 first, and then individuals 1 and 5 are then evaluated. Individuals 1 and 5 tie on that test case, then they go to individual 3, test case 3, sorry, and then individual 1 is selected out of those 2. And so individual 1 is selected. So 3 different selection procedures, 3 different individuals selected. And you can see now that this might lead to larger diversity in the population. It might lead to some interesting behaviors where there's one individual that can do something really well. So for example, back to the calculator example, one individual can handle division perfectly, but it can't do anything else. But we still want to keep this person around, even though they have zero score on all the other test cases. We want them to be around because they have this novel, important information that's important for the population to have around. Now, Lex case selection also has a very interesting environmental interpretation. So this is the exact same image. All that I did was I changed the test cases to being environmental situations that you might find yourself in as an evolving population. So we can imagine this being, let's say, 5 creatures that live in an environment, and in their lifetime, they see certain events. And these events are, some of them are life-threatening, but most of them actually affect the reproductive chances in the future. So let's just take these 4 random events. Let's say there's a lion attack, there's a tidal wave, there's a fire that you need to survive, and then there's a tree falling over and you want to dodge the tree from falling over. So it's obvious that in your population, you don't see all of these things necessarily in the same lifetime. You usually see them in a random order, and you maybe only see a few of them if you at a time. So for example, here we have all these 5 individuals, and wherever they are, there's a lion attack that occurs. And then only individuals 4 and 5 can survive a lion attack. So it doesn't matter for the population. It doesn't matter that individual 3 can survive a fire really well, because the lion attack is a thing that occurred in their life. So they need to be able to survive that to be able to move on to the next, to be able for their genes to move on to the next generation. And so individuals 4 and 5 here survive the lion attack, and then they go on and continue living. Then later on in their life, they see a fire and they need to survive the fire. And let's say they both survive the fire because they're both equally good at surviving fires. And then they move on. And then there's a tidal wave, and then individual 4 survives, whereas individual 5 doesn't. And so that's why individual 4 survived. Now you can imagine the same exact group of individuals on a different day of the year could have seen a fire first instead of seeing the tiger or the lion first. And so in this case, only individual 3 survives. And in this case, it just so happens that as a random occurrence, the individual 3, the thing that individual 3 is really good at avoiding, happened. And so individual 3 gets to make it to the next generation, just purely out of luck, you would say. And then the final one is the same tiebreaker, where there's a tree falling over and only individuals 1 and 5 can effectively maneuver around the tree. And then they're tie broken with a fire that happens later on in the life and it gets survived. So you can see here that the test cases that you're evaluated on are very similar to the environment that you find yourself in as an evolving individual. So if your parents are evolving in a certain environment with a certain set of test cases, and you're evolving in the same sort of environment, you can facilitate good evolution where you can learn traits and you can learn how to avoid certain negative things that happen. You can learn how to survive based on different test cases. So this is where downsampled Lex case selection comes in. One main issue of Lex case selection is that you need to evaluate all individuals in the limit on every single test case. And because we're doing the selection event thousands of times, we do it once for every individual in the population, we end up seeing every test case about once. And so that means that every individual needs to be evaluated on every test case for the entire generation of selection events. Now, this is an expensive procedure. And so there's been a lot of research into trying to figure out how you can decrease the amount of computation you need to evaluate a population. And downsampled Lex case selection is one such way. Instead of using the entire training set, which in genetic programming runs, which I'll get into, have about 200 test cases in, that's a pretty expensive operation to evaluate 1,000 individuals on 200 test cases each. And so what downsampled Lex case selection does is it uses 5%, 10%, 25%, or 50% of the entire training set to evaluate the individuals. So you can imagine you have the 200 training cases. You take 10% of them, and then you evaluate all 1,000 individuals on those 10%, and then you use just that 10% to select for this entire generation. Now, a way to make this comparison fair is you could also then increase the length of time you run your evolutionary runs for. So you are making computational savings, and what some people use these computational savings for is actually running for longer. So you can make a smaller, you can evaluate each individual less thoroughly, but you can run it for like 10 times as many generations. And we find here that this actually improves success rates for program synthesis problems. Really, it improves them significantly. So we can see here in green is downsampled Lex case selection, and red is the standard Lex case selection. And this is number of successful runs out of 100. And we can see that as we approach 100, they all approach performing exactly the same. So at the 100% downsampling level, all these 3 methods are exactly the same, because downsampling at 100% is not actually downsampling. So then we find that when we have, for certain problems, so maybe not all of them, but for certain problems, downsampled X-case selection has shown some promise. And this has been verified in recent work as well. So a later study by Helmuth and Spector were trying to figure out why downsampling works so well. So initially, there were 4 hypotheses for why downsampling works. The first one is that you can evaluate more individuals. So because you're running for 10 times as many generations when you're downsampling at 10%, that means that you can evaluate 10 times as many individuals, because each individual, you're evaluating them on less test cases, but you are evaluating that individual on some test cases. And so that means that you have a total larger pool of programs that you actually get to explore. The second is that you have deeper searches, and this might allow for longer evolutionary lineages. So instead of just more individuals, downsampling also allows you to have deeper searches. So you can go for 10 times as many. So it means that you have 10 times as many ancestors as you're evolved. So, and there's also environmental change, and this causes better evolvability. So for example, if the 10% of the cases that you're seeing at every generation is shifting slowly, you can actually start to adapt to the changing environment, which allows you to be better at evolving and makes you more adaptable, which makes it easier for you to generalize and makes it easier to learn. And final one is better generalization. So in this paper in 2021, Helmuth and Spector tried to verify which of these hypotheses could be used to explain the benefits of downsampled X-case selection. So not necessarily whether or not they happen, but instead whether they can be the reason that downsampled X-case selection is improving on regular X-case selection. And so they found that it likely is not the fact that there's deeper searches, because they also tried making wider searches, and they found that wider searches act exactly the same as deeper searches, or in terms of performance. And also they ruled out environmental change for a better evolvability by making a method that had no environmental change, and also found that it performed better. And finally ruled out better generalization as well. So although these things might happen, they are not the reason that downsampled X-case selection is better. And so what they landed on as the working hypothesis is that more individuals were evaluated in total. And so you have you can evaluate more in program space. So this is where the environmental discontinuity hypothesis comes in. So we think that if you're evaluating individuals on a different set of test cases than you did their parents, so let's say you evaluate 10% of the test cases on the parent generation, and then the next generation you resample from the population, or you resample from the entire training set, and you get 10% new ones. These 10% could be different to the first 10%. And so if your children are evaluated on a different environment to those that you were evaluated on, there might be some sort of discontinuity here that could be hindering X-case selection's runs. So although in previous work it was found that this environmental discontinuity did not help evolution, we're trying to figure out whether it actually exists or not. So in order to figure this out and hopefully use these results to improve our search performance, we performed some experiments. So the method for these experiments are performed in a PushGP implementation called Propeller. And PushGP is just a stack-based language that is used for genetic programming runs. The program synthesis problems we're doing come from the second program synthesis benchmark suite, which are just a bunch of introductory college level programming assignments that usually have multiple different data types, and they also require you to have looping and complex things like that. And so we chose 2 of these problems. One of them is called fuel cost, and one of them is called snow day. So fuel cost is, given a vector of positive integers, you have to divide each of them by 3. You round the result down to the nearest integer, and then you subtract 2. Then you sum all of the new integers into the vector, and you return an integer. So this problem, you have to evolve a computer program that takes a vector and returns an integer, and actually, and it does this automatically, and we only give it input-output cases. Now, snow day is a bit more complicated, and you'll see that reflected in the success rates, that snow day is given an integer representing the number of hours and 3 floats representing how much snow is on the ground, the rate of snowfall and the proportion of snow melting per hour. You have to return how much snow is on the ground after the certain amount of hours given. And then obviously, so hours are given as a discrete event, and then melting is not a continuous process. Okay, so what these 2 problems show is that they use different data type input and outputs. And so we generally trust that this is going to give 2 different views on how our methods are working for this paper. Okay, so the first method that we're attempting to use to hopefully highlight the prevalence of environmental change, and if so, use it in our benefit, is rolling down samples. So let's assume that the environmental discontinuity hypothesis is true. In that case, that means that we have jarring discontinuities between the populations that the parents are evaluated on and the children are evaluated on. So what rolling downsamples does is, instead of just re-downsampling every generation, we instead roll the downsample over evolutionary time. So you can see here in this graphic, we have this whole set of test cases on the bottom is the entire training set. And then these 4 cases is the current cases in our downsample. And so every generation, we add S cases to the back of the downsample, and we push S cases back into the training set. Now, we do this in 2 different methods. We do it in the bag method and the queue method. And so what the bag method is you remove them in random order. So you just pop them into the bag, and then you just remove them randomly. And then in the queue method, you add them at the back of the line, and then you remove them from the front. And so the results from rolling at step size of 1. So what this means is we add 1 test case every generation to the downsample, and we pop 1 test case from the population, from the downsample every generation. We can see here that we did this at a 5% downsampling rate and a 10% downsampling rate, and we're comparing to Lex case selection, which is a 100% downsampling rate. And so what we see here is, at a step size of 1, rolling with bag and queue seems to perform exactly the same as random downsampling at a 5% downsampling rate. At a 10% downsampling rate, the same result is held as well, performed not statistically significantly different to how random downsampling works. Now, it's important to note that all of these methods are significantly outperforming Lex case selection, which only has 17 success rates. So how you read this figure is that we have successes right here is a number out of 50, which is how many successful runs we had that have successfully evolved a solution to the fuel cost problem. And we have here is the max generation. So note that when we're downsampling at 5%, we actually multiply the max generational limit by 20 to actually make it so that you have the same amount of compute used and make it approximately a fair comparison. So when we have 300 generations for Lex case selection, we have 6,000 for 5% downsampling and 3,000 for a 10% downsampling. Now we see here that rolling at step size of 1 has basically no performance differences to random downsampling. So that's evidence against the environmental discontinuity hypothesis. We then varied the step size. So instead of rolling at step size of 1, we then tried this roll, and we rolled at step sizes of 1, 3, 5, 10, and 19 for the fuel cost problem, and 2, 5, 10, and 19 for the snow day problem. And again, here we see that the results are not statistically significantly different individually. So using a pairwise chi-squared significance test, we see that each individual run is not significantly worse than random, but all 5 of these runs are indeed worse than random. So we see here that this might even begin to suggest that rolling might be actually performing worse than random downsampling. So doing this method to try to smooth out the environmental discontinuity is somehow ruining search performance very, very slightly, or it could be. There's a low magnitude signal that it could be. And the same thing is found for snow day, where all 4 of the runs are performing worse than random, although individually none of them are significantly different. So this is pretty interesting results for rolling downsampling. So we made a few conclusions from these experiments, where we expected the rolling to help search performance and help us get more successes. It actually it didn't help, that's for sure, and it might even seem to suggest that it might be performing worse. So we had some reasons why. Well, firstly, when we're rolling, the first guess is that we have certain niches that are left out of the downsample for a very long time. So back to the calculator example, if we have the test case that says, whenever you divide by 0, return an error, that's 1 test case. And if we have 200 test cases, that 1 test case is likely going to be out of the population for a very long time. So if you imagine we have 10 test cases and we're rolling through the 200, it takes about 20 generations or even more than that, actually. It takes much more than 20 generations to actually get that last test case, the one that has the divide by 0 case. And so what happens here is that divide by 0 could actually be getting catastrophically forgotten. So as the population is learning the test cases in their downsample, they're forgetting the test cases that are out of the downsample. So there's a really big trade-off here between having a case long enough around to learn on it and also not having the other cases out of the downsample for long enough to forget them. So there's a balance here. And then also, finally, we also think that there's the prevalence of synonymous cases. So synonymous cases are 2 cases that, although have different input-output labels, would measure the same behavior in the population. So if we're evolving an is-even function and the computer programs that are evolving use the modulo operator and modulo 2, the inputs 2 and 4 are exactly the same to the computer program because the output is the same and the same exact operation happens in the same way. So you could say that the 2 and 4 cases are synonymous with each other because they would select for the same exact individual. So synonymous cases effectively exert no extra selection pressure when they're in Lex case selection, because you can imagine that whoever passes the first one will pass the second, and so there's no point in having the second because you can just let them go to the next test case after. And we also think that these synonymous cases might be diluting the effect of rolling. So it makes it so that the test cases, the environments are not actually that jarringly different, because inside one downsample that's random and you re-randomly downsample, you're likely to have a few cases that are synonymous for each other in both downsamples. And so the parent generation and the children generation basically have the same environment to a certain extent. That's another reason for why we think rolling might be doing not doing better as we expected, but in fact maybe doing worse. So the next step we wanted to figure out is whether it was actually due to the environmental discontinuity not existing. So we wanted to figure out whether that last hypothesis is true. So can we explain this result based on environmental discontinuity not actually existing? And to do this, we tried to exacerbate the hypothesized environmental discontinuity. So we tried to take it and make it make it worse than it actually is, and see if this would hinder our search performance. And this is how we're going to do that, using a method called disjoint downsamples. So what disjoint downsamples is we're forcing the population to have exactly disjoint downsamples. So when you're randomly downsampling, it's possible that you re-randomly sample the same test cases twice in a row. That's possible. And it's possible that you re-randomly sample it maybe 3 or 4 generations apart from each other, which could be why you're not catastrophically forgetting. But with disjoint downsample, we force you to have disjoint sets when you're actually downsampling. So you take the training set, you partition it into a certain amount of downsamples, and then every generation you pick one of those downsamples. So one generation you'd be here, one generation you'd use this test, one generation you'd use here, one generation you'd use here. And so what this does is this forces there to be an environmental discontinuity, because you have whatever cases are here are not prevalent here, and they're not here, and they're not here. And so you're forced to not be evaluated on the exact same test cases as your parents were. You will never, in fact, in this process, unless you're on the edge. So what I mean by on the edge is if you get to the end, we repartition. And so it's possible that on that last case, if your parents are evaluated on one and then you repartition and then it goes back, there's a chance that the case is there on that last and then on the first of the next partition. But that's the only time that this happens. So the majority of the time, there's no cases that are the exact same in generations that are right after each other. And so what we hypothesize is that if there is an environmental discontinuity, this would make it so much worse, because we're forcing there to be no overlap at all over like 20 or so generations. And so these are the results from disjoint downsampling, where we have at 1% downsampling rate, we see that the downsampling gets about 33 for random and 33 for disjoint. So exactly the same performance. We see that disjoint somehow at 5% is getting the same also. It's actually, if anything, getting a few more successes, but that's probably not, this is not statistically significant. And then at the 0.1 success rate, we also see that it's not statistically significant of a difference at a 0.05 significance rate. So with Lex case selection, although these are all better than Lex case selection. So still we're outperforming Lex case selection, but random and disjoint seem to not be performing significantly different. Same exact thing for snow day. We see here that at a 0.1 downsampling rate, so the same as this treatment, we see that snow day also has a not significantly different success rate between these two results. And so this is an odd result here. We see that there is no difference between the ones that are at small downsample size. But then as we make the downsample size bigger, there's also a very slight trend that's suggesting possibly it's getting worse as the downsamples are getting bigger. But this result here seems to disprove of our environmental discontinuity, because even when we try to exacerbate it, we're getting pretty much the same results in our evolutionary runs with genetic programming. So this is odd. And so our conclusions from this, which is also another failed attempt at increasing success rates, we find that actually even trying to force there to be environmental discontinuities does not significantly hinder performance. And what we take away from this is that there is likely synonymous cases in the population of test cases. And so we have, even though you're disjoint downsampling, it's likely that there's cases in the first downsample that check for the same behavior as in the other downsample. And so basically your parents are evaluated on the same behavior metrics as the children are. And so there isn't that big of a jarring discontinuity. Now, there is a slight possibility that when the downsamples are large, we actually find that the downsampling, that disjoint downsampling performs worse. And this might be just because in this case, all the synonymous cases are indeed inside the downsample. So for example, if we have 20 synonymous cases, then, if we have like 10 synonymous cases that all code for the same behavior, it's possible that those 20 are all found in the same downsample when we're randomly downsampling, or when we're randomly partitioning the training set. And so because of this, that would induce an environmental discontinuity there. And so maybe when there's a larger downsample, there is an environmental discontinuity. But what we take away from this is that there's likely the existence and prevalence, so that they're very common synonymous cases in our downsampling, in our GP runs. All right, so what are the conclusions here? And what are we going to keep working on in the future? Well, the main conclusion we can take away here is that although there is an environmental discontinuity in the training case labels, so the input and output labels, there might not be a discontinuity in the behaviors that are being measured. So in the behavior space and the things that we're looking for in behaviors, there might not actually be this jarring difference from where the parents are evaluated versus where the children are evaluated. So although both the methods we presented in this work are possibly negative results, we believe that this work highlights future directions for exploration that could yield positive results. So a first stab at this would be sampling methods that take into account case synonymy. So for example, if we could figure out on the fly whether two cases were synonymous for each other, we don't need both cases in the downsample. We only need one of them. Right? So if we could somehow use population statistics, or we can dynamically, as evolution's going on, collect how individuals are performing on test cases, we can figure out which test cases are synonymous for which, and then we can only use one of these synonymous cases in our downsample. And that's going to ensure that the parent generations are basically evolved on the same environment, or in the same environment as the children population. And this helps allow for evolution to continue and not have any discontinuities that would result in children having niches that are dying off. So an example of a niche dying off would be, again, the divide by zero test case. If we were randomly downsampling, or if we were using one of the two methods presented in this paper, it's possible that that divide by zero case is left out. And so then the children, the parents who could be really good at dividing by zero, and they want to maintain this parent that has the divide by zero test case, when we don't have the test case in the population, or in the downsample, then that person has no way to survive because they can't do anything else. And so by changing this environment, you effectively kill off a bunch of niches of solutions that we want to maintain as Lex-case selection. And indeed, Lex-case selection actually does do really well at maintaining specialists, but that's only under the condition that the cases that the specialists are specialists on exist. And so if you don't have those cases in the downsample, then those specialists can't survive. And so what we want to do is ensure that these specialists can survive and they can make it to the next generations. And to do this, we need to make sure that these specialist preserving cases are in the downsamples. And yeah, so I'll be happy to take any questions now.
Great. So do we have any questions from the audience?
What would you consider a true jarring difference? Oh, that's a very good question. So I would consider a jarring difference to be something where it's the difference between the environment that the parents are evaluated on and the environment that the children are evaluated on to be something that makes the children seem not fit when they should be. So for example, if you have children that are really good at doing certain test cases and doing certain behaviors, but those test cases aren't there, you're going to say that those children are bad when you're trying to select them, right? You're not going to select those children. And so that would be a jarring difference because we're losing these things that we worked so hard for. And so we worked very hard to build up these behaviors, this specialist repertoire. And if we're not having these cases that are really important to have, then we lose those specialists. And so that's what I would consider a jarring difference.
I see. Thanks.
Of course. So I see that Lee has a footnote. If there's not any other questions.
Yeah. Oh, maybe people will think of questions, but I'll just note. I don't think it was probably clear to everybody in this audience that every method in the presentation is significantly better than what most people do in genetic programming, which is tournament selection based on total error. So the vanilla Lex-case selection that we saw being, oh, that's much worse than all these downsampling things. Well, that's much better than tournament selection based on the total error, aggregate average error, which is what most people in problem-solving oriented evolutionary computation do. So doing the random permutations with the sort of environmental challenges in random orders is a major boost on many kinds of problems, most. And then an MSU team showed that, downsampling even better. And then all of this stems from trying to figure out, wait, why is it better? Can it be even better than it is? And there are all sorts of sort of ecological considerations that we're wading into, trying to figure out what's going on with the environments in which the parents are being selected. And I guess I'll just, it's a long footnote. I'll also say I am super grateful for the organizers of this workshop, because these questions are better than the questions when it does work, right? Like we definitely had all sorts of assumptions about what was going on that would lead us to think, well, if we do this, it'll certainly have this effect. And then when it doesn't, you really have to question your foundational assumptions, and having a target for it is great. I think it's one of the best developments I've seen in any of these associated fields in a long time. And so I want to applaud the organizers and hope this happens more.
We are grateful for your contribution. If there are any other questions, we can take questions. Otherwise, I think one of the things we wanted to discuss generally, I guess, following on Lee's comment, is like, how can we get more people to participate in this kind of research? I think it's probably important to acknowledge that a lot of times it's actually more work to investigate these hypotheses than it is to just do something and like it either works or it doesn't, and then you move on to the next idea. So I guess for the people that are here in the audience, like what could we do to encourage you to do this kind of work and investigate negative results, or I don't know.
So my only thought about that is, when was this first announced? Maybe it was announced with plenty of time, but my only constructive thought maybe is more and earlier publicity, because people generally drop things when it looks like it's not panning out. And if they already have implanted that there's actually a venue for it, early on when things aren't working out, they realize that's a possible outcome, which usually isn't in these fields, right? And there are reasons, but often you learn a lot more by pursuing this kind of why it didn't work kind of question. And I would say the more people can be conditioned to know that that is a possibility as negative things start to happen, some of them are going to end up with nothing worked, but we learned something. And often people's documentation peters off before, right? If it's really not working out, they're not going to be as rigorous. But if they know, oh wait, there may be a reason to keep really good track of this. So I would just say earlier publicity, louder publicity. Maybe it was early and loud this time, but I remember we found out about it a little bit later and we're like, wait a minute, that might be good. And it was just at the right time for this project.
I think we started publicizing it maybe at the end of February, somewhere around this time. I think also maybe one thing that might impact the number of people who submit is maybe some people just don't have the confidence that they did something that actually made sense, and they think, oh, it didn't work because I didn't have the right idea, or like people will think it's stupid because it was obviously wouldn't work or something like that.
Yeah, I think that's a really real concern. When you get a positive result, it's easier to convince yourself that you didn't have bad assumptions, and especially when you're writing code and building a complicated system, and it could just be some really dumb bug, right?
Right.
I would say actually for us, it was probably that we were interacting with people outside of our group also who were working on downsampling I think maybe Ryan, you can help me think that through. But I remember thinking, well, we know that this is not a crazy idea because of these other conversations we've had, not just amongst ourselves, that, you know, maybe making the down samples less discontinuous should matter. So it wasn't just a weird idea of our own.
Yeah, I think because we were forced to explain the ideas when we were talking to the other group about it, that forced us to already like write them out and think about why we did it. And then in doing that, it was a really productive exercise. And then we also learned that they also tried very similar results. And so if we had this workshop 1 year ago, maybe we wouldn't have had to do that. So I think it's a really good idea. And I honestly think that we do gain a lot from publishing negative results, because no matter what, you're going to learn something either way, whether you're right or wrong. So it's productive in both senses.
I see we have a raised hand from Tanner. So Tanner, if you want to go ahead and.
Yeah, briefly. I'll try not to wax too poetic about it. And part of my thought here, I'm coming from industry into academia, but I have a background in studying failures on a sociological level. And a large part of what I see makes a big difference it's training and it's experience and practice with it. As we've seen with the 2 talks that were given, there was some rigorous effort that went into understanding why there was a failure. Right, that's a thing that can feel dicey. Is it just because the code was bad? Is it because I had some bad assumptions? If I chipped away at this more, would there be something else? And there's also this fear, especially among those who have had good training as scientists, to be like, I don't want to paint with too broad a brush and assume something incorrect based on I don't want to generalize the failure too broadly. And so training and practice in performing this sort of activity and learning to see past hindsight bias, but also understand that when something goes wrong, you are getting a mental model update on how your system actually works. There's a divergence between how you thought your system worked and how it actually does. And then that's a good and valuable thing that we're looking for. It's just not the shot that you called, so to speak. As the community builds up and we can spend time teaching each other how to do this, that can develop a critical mass. But I found that it's a 1-by-1 chain reaction sort of approach where you work with and you mentor people and you teach them how to do it, and then they go teach other people how to do it. Maybe also small workshops. It's not really like a broad lecture-based thing people pick up. It's a mentoring thing, but it makes a big difference. And once people buy into it, they really love it and they really get it.
Maybe both of those previous comments suggest that something throughout the year where people can consult each other on negative results. As we had some of that with a pre-established collaboration that gave us the confidence that it wasn't a crazy idea or a bug. If there is some sort of network of mentorship and collaboration where you would go for things that aren't working to help think it through, and whether the idea really was something general that people would care that it didn't work, and so on, would be a good idea. It's more work, but maybe that would help.
Hiroki raised his hand.
Yeah. So hi everyone. So thank you very much for organizing this session. This is a fun session. We should see more of stuff. So my feeling about initially when I saw the call, I thought there's going to be a really fun stuff. I should submit something, but the required level of rigor was a little bit too high, so that we are supposed to explain why something didn't work, right? And I'm sure that there are many people who don't even know why it didn't work. So they may want to share their experience here without much rigorous analysis and try to get feedback from the audience. So if we loosen up a little bit of requirement for this workshop, I'm sure there are more people who want to submit something. And especially fields like ALife, there are so many open questions, like how to achieve the consciousness, how to achieve the open-ended evolution, how to achieve hierarchical emergence. Every single result so far is negative result, and nobody actually knows why. So yeah, things like that exist in many different places. So if we don't require specific explanation, but maybe some kind of hints, maybe this was not quite right, etc. So if we loosen up the requirements, I'm sure there will be more participation. Yeah.
So all of these comments suggest to me that maybe we should have a workshop that is in two parts, like the rigorous we did put some hard work for a year part, and then the part where people are just exchanging about stuff that they did that they didn't work and kind of ask the community whether it's actually interesting, an interesting failure, or if it's like something not so interesting. Any other ideas? I think maybe if we have for next year, that's probably what we should do. And in preparation for next year, if we want to do this, we should probably have an event like really soon after the conference for people to just discuss like failed experiments and start working together, maybe mentoring some students, stuff like that.
Perhaps we can send out the call for submissions right after the acceptance rejection notice of the main conference.
Oh, very smart. Very smart timing.
Yeah, I think that thought occurred to me, but I think maybe that comes back to the level of rigor thing where there wouldn't be enough time probably to do like this sort of big systematic study, unless it's for like, the next year or something. I agree with basically everything everybody's said. I think there's some really good suggestions, and yeah, maybe we can have like kind of two levels of this workshop. And then, yeah, it's like, oh, got a rejected paper. Let's come figure out why. And that would still be, I think, in the interest of like furthering the science. So
Maybe one part is called the "it didn't workshop," and the other one is the "why it didn't workshop." Maybe somebody can go with something better, but something else to sort of, the why is the heavy part, I think, that Hiroki was talking.
Right, it's true that we had a very high bar for submitting the paper this year. I guess we were a bit worried that we would get a lot of papers that said this didn't work and maybe didn't work for a trivial reason, but we didn't even investigate why. Especially because the organizers were the reviewers, so we did not have a lot of reviewers to count on.
I think having some good examples as a model is a great place to start, certainly. And I think that was valuable. But yeah, I think having the degrees is the next logical step. But starting with, I don't know, North Star is probably too dramatic, but with these two really good examples, it helps people to conceptualize what they might be able to get to going forward. And yeah, demonstrates a little bit of what some of the value of this rigorous activity would be.
yeah, it probably also just helps since we're talking about how far in advance do we need to publicize and things like that. Like this was the first year that it runs, but if we run it again, which I would like to run it again, now there's like an established precedent that like, oh, okay, now we know that next year there will probably be this workshop for doing negative results, so maybe people can be more aware ahead of time.
If there are no other comments, maybe we can free people to go see I think there is the keynote happening maybe right now.
Oh, is there? Yeah, we should check that out if so. But yeah, I just wanted to say thanks again to everybody who participated, and we hope you enjoy the rest of the conference.
Thank you, everyone.